Namaskar दोस्तों, welcome to channel Toxicology Abroad. Today in this video we'll be discussing another DABT topic. We'll be discussing in this video. I have appeared for the exam in the year 2023. So based on my memory, I have made a list of whatever I could remember. What are the topics which we came in the exam? So I have shortlisted these questions. Frankly speaking, it is very hard to remember once you come out of the exam because the exam is for 4 hours. Once you come out of the exam, to remember exactly what were the frame of questions, to remember it is very challenging task. I came out and I was traveling back to my home. I was in the bus, so immediately in my notes I tried to remember what were the questions, and I have listed. Anybody who have appeared for the exam in the year 2023, they can also give in the comment section if they remember any questions. So I'll quickly go through the questions the concepts which I remember. So first question which I remember was one of the questions which I remember was the concept what they have asked me with respect to physicochemical properties, pH, pKa, ionization of the drug, absorption in the stomach, small intestine. The question was related to this concept. If you look into ABT Brochure 2026 also, it is the part of syllabus. It is there, see, considerations for measuring the appropriate constituent, example, parent versus metabolite, that is unbound, free or total. So there is something called Hasselbalch equation will be there. If you remember in the case of kinetics also, so always they'll ask pH of the drug is this much, in the stomach this much is there, how much is absorbed, how much is unabsorbed, what is the ionized, what is the ionized drug, what can be given as an antidote. So relating to these concepts, even for pharmacologists or for the formulation scientist or for the toxicologist, they keep asking, the examiner will keep asking. So this is the primary concept that you have to remember. So see what are the examples are there. Sodium bicarbonate might be there, some antacids will be there. So remember that one. Try to read the examples. Go back and check in the Clayson also if these equations are there. Did they cite any example drugs are there, sulfanilamide, or they might have given some antibiotics or some antacid drugs they might have given. So see the examples and try to read it. Because exact question I can't remember. I can't remember as of now. Next one question with respect to route of administration they have asked. That is, they might have given x root greater than x root greater than x root like that. So there is a comparison was there. Intravenous, subcutaneous route of administration, the bioavailability. So they might have given some example and the comparison between IV and subcutaneous more, which of the chemical will give more bioavailability like that. And one question with respect to transdermal route. So even if you see ICH Q3D guideline also, there is a recent revision is there like 1 or 2 years back only, including percutaneous absorption. So transdermal drug delivery, how to include that absorption factor with respect to the correction factor with respect to dermal absorption, those things are also very important. So examiner also included this type of concept here, transdermal route of administration and bioavailability. So routes of administration, it is there in the ABT brochure. There is a comparison also you can see in this section, metabolic differences between various routes of administration. Example, sublingual versus oral. Here what they are given, comparison between intravenous and subcutaneous. So like that you can check in the ABT brochure. So another concept question which was across the concept, mutagenic impurity. So ICH M7 guideline you have to read. So if you look into ICH guidelines, safety guidelines are there, quality guidelines are there, multidisciplinary guidelines. So in that they will ask you one question with respect to M3, M4 or M7. So M7 is the one which is related to mutagenic impurities. So certain class of chemicals for which there is no literature is available, by default you can go, for which you cannot derive health-based exposure limits like permitted daily exposure. You can mitigate the risk by using a default factor called TTC, 1.5 microgram per day. And you have to read with respect to ICH S9 guideline also. There are certain drugs which are used in the treatment of late stage cancer treatment patients, for which the treatment duration is very less. There you can mitigate the TTC level to much more higher level. Okay, so that also, that table, ICH S9 table you read. In my channel, I have already covered ICH S9 guideline related question and answers. I have already covered. In that, I have also discussed treatment duration, what are the TTC values. So please refer to that. I will give the link in the description. Then one question with respect to ICH S6 guideline. S6 guideline is something related to biologics or biotechnology derived products. So most probably what they will ask regarding this particular concept is relevant species. What are, if I am developing a biotechnology, biologics, what are the toxicology studies need to be conducted if I am filing for FDA or certain regulatory body. So you need to be aware of this ICH S6 guideline. So S6 guideline related two videos I have already uploaded in my channel. Please go through what are the relevant species, what are the toxicology studies. They have given certain tables also. So read that one. Next question, something related to QSAT they have asked. That is, since now new recent guidelines or updates are more towards read across or advanced outcome pathways or something related to QSAR or in silico tools like Derek, Lassa or Sarah. These are the softwares where considering 3R principle, regulatory agencies are stressing more on the industries, whether it is chemical industry or pharmaceutical or cosmetic industries. They're pushing, they're encouraging these scientists to go for alternative methods. So same kind of information, if you can get it from using the software to mitigate the risk, earlier they used to wait for in vivo data, but now by using for example for the qualification of the impurities also you can still use direct you can use in silico prediction and you can mitigate the risk. You can prove that the safety of that particular impurity. So like that, one question was related to QSAR. So QSAR similarity. So read across is very important. That functional motifs, toxicophores that you have to consider, molecular weight. So this is also very important. Molecular weight correction factor. You are working on a certain X chemical, and that X chemical is added some methyl group or some chloride or some functional group is there. So how the addition of that particular CH3 group or OH group, some functional group, how it is, whether it is increasing the toxicity or it is is it similar to that of the parent molecule, like that. And if there is any change, how we can apply molecular weight correction factor, that you have to see. Salt form case study, something related to dismethyl. So there is a metabolism might be there, or some methyl group was removed from the actual drug. So how, whether we need to do a fresh new toxicology study or we can mitigate the risk by doing molecular correction factor, like that one question. That was something calculation question was there. So like that they will ask. So another question was with relative to RFD. So in the case of US regulatory agency, you have this US EPA RFD oral reference dose and inhalation reference concentration RFC will be there. So similar to that of health-based exposure limits. So similarly, we have RFD will be there. So based on NOEL or BMD, considering as a point of departure, you'll be calculating this RFD. So mostly, repeatedly, if you see previous ABT recertification questions also, they will ask one question with respect to uncertainty factor. In the option 10 into 10, 10 into 100, 100 into 10, like that, they'll try to confuse you. So mostly the question area is related to uncertainty factor 10 into 10. One of the options will be like that. So remember, so if you look into the ABT brochure, so in this section F, there the concepts and use of safety threshold, example TTC, Kramer class, reach DNES. So if you look into ECA reach registration, no DNES, that is derived no effect level, and DNES reference values, that is RFD, acceptable daily intake, that is WHO will give you, and health-based occupational exposure limits that you will do with respect to worker safety. So like that, health-based questions, something related to health-based exposure limits, they will definitely ask in the exam. Some questions with respect to data-derived extrapolation factors or correction factors or uncertainty factors or cumulative safety assessment factor, like this. Some questions related to that they will ask. Or something called child for children, no additional 10 used as a uncertainty factor. So questions related to that concept also they will ask in the exam. Okay. So another question which they ask with respect to circling superfund. So in my channel also I have already US related regulatory agencies with respect to soil, air, water. I have already uploaded in my channel. Please go and read, because these are theory based questions. If you know what is this particular organization, what is the agenda, what is the objectives of this organization, you will definitely know. So read about what is circular and superfund. The main purpose of this is cleanup goals. For example, there was a in Europe, if you see, there was a seawater disaster was there, or in US only certain areas some chemical spillage is there, or in nearby ocean near the America, if there is any water what is it oil spillage is there, accident is there, how to clean up that particular area, because nearby aquatic animals should not get affected. So cleanup goals is important. Who is responsible for the spillage? Who is taking the responsibility? How much time it is there to clean that particular? It's not that you try to clean it from 1 year to year. It has to be effectively implementation should be there. So cleanup goals, emission limits. If the factories are having some emission limits, safe concentrations are very, very important. So one question with respect to US regulatory agencies, they've asked, that's why. In addition, you read about air, water, soil. Okay. I will give the link in the description. Next question with respect to OSHA, that is occupational safety hazard and permissible exposure limit. That is actually what has happened in NIOSH 2024 also latest list of guideline chemicals as well as their categories they have given, their limits they have given. So read NIOSH 2024 list. What are the potential chemicals which are having hazardous um, there's a table 1, 2, 3 they have listed. So read that one. That's why there's some questions with respect to latest new guidelines updates also they will ask in the exam. Okay. So OSHA PEL, various regulatory bodies which are responsible for establishing occupational exposure limit. So those who are working in the risk assessment area, you might be well aware of this particular formula. Pause the video. Those who are not aware, you can go through. And if you need any explanation how to calculate, you can give comment. I will explain you one case study how to derive occupational exposure limit. The next one question with respect to PBKK modeling or pharmacodynamics. So in addition to toxicology or TK modeling, some questions with respect to pharmacology also they'll ask. So here the question was with respect to PK modeling. So if you look into ABT Brochure also, outputs of physiologically based pharmacokinetic, that is modeling, and their utility in characterizing dose response relationship. So in the first four chapters of Clayson also, if you see principles of toxicology, this PPPK modeling is very, very important to understand dose response relationship curves. From this only you will be able to derive the EC50 values. So for that reason, this is very, very important. Try to read the concept behind this PPPK modeling. Okay. Next, some questions with respect to TiA2, that is nanoparticle bioavailability, surface area absorption. The basic concept that we have to remember is as the surface area, as the particle size decreases, surface area increases, absorption increases. This is the concept. But with respect to nanoparticles, since the particles are too very small in size, aggregation of particles might take place, which might increase the particle size. So read those concepts. Okay. With respect to nanoparticles also, I have uploaded one video in my channel. I'll give the link in the description. Please go through, read about nanoparticle toxicology, examples also TiO2, colorant might be there, zinc oxide is there, metal oxide is there. So read these examples. So standard parameters used to predict to describe difference in localized and systemic toxicity. That's ADME, PKP, that is pharmacokinetics, toxicokinetics, pharmacokinetics and pharmacodynamics, parent versus metabolite. So from this you can understand here this concept is covering this pharmacodynamics and PBPK modeling. It is also covering under ABT brochure. So these things are taken from ABT brochures. You can go back and check section F, B, F, A under various domains. Okay. So one question with respect to ISH M4 guideline. As I told you, with respect to multidisciplinary M3, M4, M7, they will ask. So here one question M4, I think it is with respect to ECTD. Those here will be there. No, when you are filing for ANDA filing. So you will fill certain CTD common technical document you will be filling. So for that various sections will be there. So these data they have asked pharmacology, toxicology, PK studies need for regulatory submission. That's why I have uploaded some videos with respect to filing also. ANDA filing, what kind of studies needed. So please go through that also. Some acts also, IND filing also I have also read type A and type B. FDA meetings will be there. Those also, what are the outcomes, pre-IND meetings, what are those pre-manufacturer notification, PMN, that also you read it. Okay. So next we will see what another. So one question with respect to statistics also they have asked in the exam. Always, if you look into historically, if you see few questions on statistics, always in the ABT they will ask because data is important. Application of the statistic, making it statistical significance, biological significance is very important. So elements of scientifically valid hypothesis-driven research, regulatory guideline study, including sections on statistical analysis. So it is a part of ABT brochure. So read what are the different tests are there. Okay, how you are going to decide the group size, what kind of type of statistics need to be applied for what type of study design. So like that they will be asking in the question. Okay. Next, one case type of question they have asked. So the question was, there was a child who was consuming applesauce, okay, and that applesauce contains some food additive, and they have given child body weight also, and they have given that child is consuming this much consumption of this applesauce meal, and they have asked us to calculate the food additive concentration, something they have asked. I spent too much time on this question, so like that, some calculation related questions they will keep on asking. So try to read some what I think here the concept is you need to calculate acceptable daily intake. You need to also read what are the direct food additives and what are the indirect food additives. So I have discussed about those things in my channel. I'll give the link. Please go through that also. Then next we have one question with respect to immunotoxicity they have asked. So ICH S8 guideline is the one which is in relation to immunotoxicity. If you are developing a certain drug and that drug is triggering small molecule drug, we are not considering biologic drugs, non-intentionally, unintentionally, if that is going to tweak the T cell helper cells or it is going to trigger the immune system, whether it is either causing immuno enhancement or immunotoxicity. So for those type of studies, you have to refer to ICH S8 guideline. So in the exam, they have asked one question with respect to. So currently, I have covered around two videos with respect to, yeah, three videos I have covered with respect to toxicity, ICH S8 guideline. So please go through that. I will give the link in the description. Still three more videos I will discuss with respect to ICH guideline, because I want to understand the complete guideline A to Z, that because any question with respect to ICH guideline comes in the exam, you will be able to answer. Okay. Next question with respect to the different types of toxicology is there, no? So try to read, try to understand. Like mechanistic is there, descriptive is there, analytic is there, forensic is there. Try to understand what is the concepts behind this type of the different classes of. Even though a toxicologist might be working in only certain toxicologists might be only GLP toxicology, certain might be only inhalation toxicology, certain toxicologists might be only risk assessment toxicology. But the exam which you are preparing or which you are appearing, ABT, that is Diplomat of American Board of Toxicology, no, it is a general toxicology. So they want to overall assess you as a, even though your work area might not be mechanistic, still the questions with respect to mechanistic toxicology, the concepts which is related to mechanistic toxicology, you should be aware of. In the exam, they will definitely ask. So read about those things also. Something related to analytical also they've asked. That is FTIR in carbon labeling and this M by Z mass spectroscopy they were asked. Frankly speaking, I did not read for the exam these particular areas because one question like if you look into analytical toxicology or forensic toxicology, no, once you get the samples, you have to analyze and you have to interpret that analysis, whatever the results the peaks and graphs are you are getting, no, you should be able to interpret and analyze. No, as a toxicologist, that is also your job to understand it. But not all toxicologists might be working in this area. I am also not working in this area, so very difficult. But questions they will ask, as I don't know, M by Z mass spectroscopy or FTR, carbon labeling, they have asked in the exam, they will ask. Okay, read it. If you get a time, try to read it. Then next to some questions with respect to inhalation toxicology, they have asked. That is, you have to understand whenever you're reading major organs anatomy, you have to understand. So in the case of respiratory system, this type of cells very important. Alveolar macrophages, alveoli, type 1, type 2, Claral cells, pneumocytes. So go back in my channel 2-3 videos with respect to inhalation particle size, the anatomy I have discussed. Try to see, or you go back to Clayson or try to read first two pages. If you there is a inhalation toxicology chapter is there, try to read this type of different cells. Okay. So one question with respect to hypersensitivity. So skin sensitization or hypersensitivity, it is a favorite topic for this examiner. They keep asking either GPMT or LLNA essay or ear spelling, MEST essay or HPRT essay or ECCVAM, that is, what are the alternative methods for identifying skin sensitization? They'll keep asking. Four types of hypersensitivity, they'll keep asking. So that's a favorite topic for these people. Read. They'll give you some examples, some case studies, and I'll try to ask you what type of either it is IgA or IgE, IgG, like this they will ask. Okay, whether it is immune mediated or cell mediated or acquired immunity, like this they will ask. Next, something related to whole body temperature was there. I also didn't understand. Maybe that is a parameter will be there if you conduct the repeat dose toxicology study. Rectal temperature or something like that they might have asked. So another question what they have asked is with respect to some they have given some the pesticide is there. The pesticide has been um, registered in the European region and they have a NOAL for that particular study. So if they are using that pesticide in the case of US, country is changing. So will they need to again repeat the toxicology study, something like that they have asked. So we'll quickly go through once whatever I remember. I am going to read it out. So ICH guideline, NOAL available already. Okay, for EU registration of the pesticide. Do we still need additional repeat dose toxicology study to establish NOAL, or can BioWare be done? So like this BioWaiver or bridging studies, like this concept related questions also they will ask. Okay. Next question they have asked with respect to content. There was a certain river body there where certain chemical has been polluted. Contamination of chemical X in a nearby river need to calculate safe drinking water. So actually I'm trying to get that particular formula. I think CEDA some formula will be there. Big formula will be there. I remember I have some examples, but I'm not exactly finding it now. In the subsequent videos or in the comment section, I will give you. So, like this cumulative assessment or safe drinking water, like how we have health-based exposure limits for drinking water, also you have some limits. So that formula they were asking us to calculate. Like this, they have given this comes under risk assessment only. I don't know how I have answered this question. I don't remember exactly. So, but they have asked. So with respect to drinking water, whatever the guidelines are the US SDWA, the Safe Drinking Water Act, you read it about it, and I'll give that formula in the comment section. I'll find it and I'll definitely give it. I'll share with you people. Next question with respect to acute toxicity study, that is what they have given is 14-day repeat dose toxicity study is already available. We need to calculate safe limit, that is HBL. Okay, dose of a chemical X options where based on QSAR can be mitigated. See, generally for calculating any HBEL or permitted daily exposure will be there, no? Generally, the guideline recommends is at least 28-day toxicology. Then only you can understand how the drug is acting on repeated administration. 14-day duration is very less comparatively. If you do not have any other information, you can consider the NOAL from the 14-day study also. Okay, that's why I say it's a case by case. So here they're trying to make us tricky. So they are saying 14-day study is there, NOAL is there, and safety limit was established based on. So now there is an ongoing 28-day study or 90-day study. Should we wait for this completion of this 28-day study or 90-day study and wait that NOAL will develop from this study, and that should be considered as a point of departure and derive the health-based exposure limit? Like that they're asking, or you will consider 14-day study and immediately stop that particular population for consuming water. So they have linked this question to the contaminant which is present in the drinking water. So we need to calculate the safe limit. Dose of chemical X options were based on QSAR options. What were based on QSAR, can we mitigate the risk, or should we wait for 28-day, 90-day study to generate the tox data? What is the immediate solution? Consider 14-day and calculation of the safe dose. Stop population for consuming water. So tricky question is there. So see how is based on options. I am not remembering exact what were the options. So like that they will ask, keep asking in the exam. So these are the things which are taken from ABT brochure only. Standard US and global non-clinical testing guidelines for pharmaceuticals and chemicals. Example GLP, OECD, US EPA, ICH, US FDA and EMA. Develop or provide clinical treatment recommendations for poisoning incidents, including antidotes. So with respect to whatever, they will give you some kind of poisonings in the options. They will give you antidotes. So like that, so you should be knowing mechanism of the toxic action. You should know how it relates to treatment of poisoning, including physical measures, decontamination procedures. Nowadays, whatever the FDA inspections are taking place, no, they're asking for the decontamination procedures. So in the case of analytical, these things are very important. Analytical toxicology related questions, these are. Then decorporations, clinical treatment options, example, antidotes. Okay. So another question with respect to poison ivy. I told you, no, they'll ask you what is the hypersensitive type of hypersensitive reaction with respect to poison ivy, skin sensitization, Urshiol. So one video I've already uploaded, please check it. Repeatedly they have asked in the previous ABT exams also. Next one question with respect to, they have given a study design. They asked us to calculate the test item. How much of the test item is needed to conduct 28-day toxicology study, like that they have asked a typical as per 408 guidelines, something like that. So whether you will consider recovery group, whether you will consider um, TK group, what are the parameters based on that you have to be calculating, no? So that question they have asked. So who are the GLP study director are there? No? For them, this area is very easy for them to answer it. Next, one question with respect to ICH S9 guideline they have asked. So, so whatever the new pharmaceuticals which are coming in the market currently, no, new type of FDA approved drugs, most of them are cancer drugs are there, anti-cancer drugs, and most of them are either ALK inhibitor or TGF inhibitor, something like a new area, which are mostly biotechnology derived products are there. So, and most of these drugs are used in the late stages of treatment. The patients who are and the end stages are there. The patients who can survive only next 1 year or couple of months. So for them, this ICH-9 guideline is very, very important. There is some relaxation with respect to TTC approach also. That is based on treatment duration. That's why it's a new revisions have taken place in the year 2023 or 24, 25 also in ICH S9 guideline. So examiner keep asking questions on ICH S9 guideline also. Next, we have one question with respect to disulfiram, alcohol poisoning. They've asked disulfiram as an antidote, mechanism of action, and abuse. As I told you, know, it is covered in the ABT brochure. So, in the video, in our channel, 5 to 6 videos with respect to alcohol poisoning I have already uploaded. Please go through what are the mechanisms, what are the enzymes, that is alcohol dehydrogenase, acetaldehyde dehydrogenase, what is a toxic metabolite is formed, what is the mechanism of action, how comparison of all ethanol poisoning, methanol poisoning, rubbing alcohol, that is isopropyl alcohol poisoning, and ethylene glycol poisoning also. So they are also metabolized by alcohol dehydrogenase. So antabuse. In the case of methanol poisoning, how ethyl alcohol is used as a antidote for the treatment of methanol poisoning. The formic acid which is formed in the methanol poisoning which causes ocular toxicity. So all these areas, no, they keep asking in the exam. So ethanol cannot be removed from our society. It will be there. So questions with respect to ethanol poisoning, you will keep getting in the exam. That you have to keep it in mind. Okay. Now, though, you can see that in the vehicles also, people are like E10, E20 or E30, alcohol mixed in the case of petrol also. So ethanol will not go. So questions with respect to ethanol poisoning definitely come in exam. Next question what they have asked is arsenic, some kind of salt. So metal related questions they'll ask. So very deeply I have given the classification of the metals also, minor metals, major toxic metals. So arsenic comes under major toxic metals. So arsenic, some kind of salt they have given, and they try to question the frame with respect to read across. If so and so arsenic salt is there, how you will do some kind of risk assessment? The question was related to the like that. So when you're doing literature search, no, on that specific chemical you will not get, on the specific metal you will not get. Always you will get it in the form of metal form. So then you have to apply some molecular weight correction factor. How you can consider salt in the place of actual metal for the risk assessment, that you have to explain, that you have to keep it in mind. So one question with respect to bioterrorism they have asked. That was very shocking for me when I first saw it is in the exam. So options were with respect to tetrodotoxin, puffer fish you have to remember, botulinum toxin, botulism, E. coli, and sarin. Sarin which was used in the case of World War II. Oxygen, saline, chlorine, these are the examples. So in our channel, we have already covered tetrodotoxin, botulism, all these things. So I'll give the link in the description. Please go through in detail. So bioterrorism is something new word which I came across. Okay, that is not covered under ABT brochure, but socio-economic or disasters, those things are already, or some zoonosis or infectious disease that will be mentioned in the ABT brochure. So if that comes under bioterrorism, I don't know. But warfare gases or the gases which are used in the World War One and Two, that are part of the ABT brochure. That is there in the syllabus as well as it is there in the Clayson textbook. So next question, something related to hormones they have asked. Uh, that is TSH, FSH, pineal glands and adrenal glands. Try to understand endocrine disruptors is a new favorite topic for the ABT examiners. So lot of new revisions, endocrine disruptor screening has been added to a lot of OECD guidelines also. So they keep asking the endocrine system related questions. They'll keep asking in the exam. So try to read it in detail. Next, some questions with respect to blood-brain barrier they've asked. So always they will try to give you some example which are able to cross the blood-brain barrier or blood testis barrier or blood placental barrier. They'll try to confuse you. So read all these barriers in our human system anatomy, and also if there are certain examples, try to read those. For example, alcohol is there. There are certain organic solvents are there. Blood-brain barrier is a tight junction, but still these alcohols or these organic solvents, no, because of their lipophilic nature, they can cross the blood-brain barrier. That's why if you see all the solvents, they cause CNS toxicity or neurobehavioral effects in the case of workers. So that is the reason why physicochemical property of the that particular chemical is very important. Lipophilicity. Okay. So read that concept that you have to understand. Next, with respect to biomarkers, they keep asking whether it is liver biomarkers or kidney biomarkers. In my exam, they asked with respect to liver enzyme biomarkers, ALT, AST. Kidney also they have asked KIM1, creatinine, total protein, they have asked. Another thing is with respect to cancer, no, they have asked biomarkers only. That is suppressor gene is there, no? P53 suppressor G or BRCA. So Clayson, there is one table is there. I will try to give that in one of my video. I have already kept that table. What are the genes which are responsible for this tumor suppressor? No. So what we do, what we as a pharmacologist or toxicologist know, we only try to remember only B53 or BRCA. In the options, there were some other things were also there. So what are those I will give you? Okay, I'll give the link in the description. I think one of the video I've already made with respect to tumor suppressor genes, that list. Next one question with Buller test. As I told you, LLNA, GPMT test, skin sensitization test, whatever is there now, MEST, SA, HRA, PT, or so, all these things they'll keep asking. Next question was with respect to TI, therapeutic index. So calculation of therapeutic index, margin of safety, margin of exposure, or HED, they'll keep RFD, they'll keep asking in the exam. So what is this TD50? They'll give you either TD50 or ED50, they'll give you or ask you to calculate TI, or they will give you therapeutic index and they'll give you options. And options will be all be nearby only. You, because why I'm saying this, it will give you TD99 by ED99, TD1 by ED99, or TD1 by ED91, or like that they will give you. You'll feel that all the options are nearby options only. Even though you know the right answer, by sitting in the exam, by looking at that options, no, you'll get confused. So you, you're in this like this type of repetitive concepts, you should not get confused only. Margin of safety ho, RFD what a formula is there, or TI formula is there, that should be impregnated in your brain. Even though options are there, you should not look at the option, get confused. Okay, you have to by default you have to pick the right answer. Okay. Next question with respect to occupational exposure and OEB, they have asked something related to personal protective equipment. As I told you, recent changes are there in the case of NIOSH. That's why they're emphasizing more on the worker protection. So probably they might have some worker accidents, they might have taken place in the recent. So that is the reason these examiners are focusing on PPE related some questions also, like hazards, symbols like that they will be asking. As I told, hydrophilicity, lipophilicity of the chemical, physicochemical properties is very important. Next question with respect to bird oral gavage administration, they've asked. So ecotoxicology, if you look into ABT brochure, no, three guidelines with respect to 200 series, OECD 200 series, I think 201, 23 or something is there, where you can calculate the bird toxicity also very important. LC50 or something will be there. Daphnia fish toxicity will be there. So read those things, three guidelines. I am not a ecotoxicologist, but in future I'll try to make three videos on OECD, the guidelines under 20123. For some summary or some notes, I'll try to put it so that it will be easy for me as well as for you people to read it and understand. Those who are not ecotoxicology, those who are working on ecotoxicology, preparation of this PC, PEC or PNEC concentrations will be there, no, for ecological risk assessment. This is their area, they can easily answer this question. Next, some questions with respect to carbon monoxide poisoning. So always methemoglobinemia, cyanide poisoning, carbon monoxide poisoning, or that is what is it, another methylene chloride poisoning, dichloromethane, paid strippers. It is a way because in America a lot of accidents with respect to these things keep happening. Closed fire will be there, where carbon monoxide will be there, something will be there. So this is the examiner's favorite area of topic. So they will ask. So you have to see blue cyanosis, all those things. What are the hallmarks are there that you have to remember? Okay. Next question with respect to hazard index and hazard coefficient they've asked. So it is a part of risk assessment only. Very important in my channel. How to calculate this H and HQ I have given with case example. So I'll give the link in the description. Please read. Very important, but you have to remember the formula HIHQ. What is the formula is there? You have to remember. They might give you some values and they'll ask you to calculate hazard index. Next, amphibian larvae, fish larvae effect of hormones. I told you know with respect to ecotoxicology, environmental toxicology, that OECD 20123 guideline, PEC, PNEC, that we have to calculate BOD, biological what, oxidation demand, BOD, that water contamination that they will ask, and effect of hormones. So because of these hormones, no, the larvae instead of developing into female, I think they will develop into male. So something related to this that is going to imbalance the ecological imbalance. No, more females or more males will be formed. No, that dysmorphism. I don't know what is the term called. So they will ask one question like this. Okay, gender dysmorphism will be taking place. Larvae will turn into some male or female. So related to that concept, they'll ask in the exam because of the chemical contamination or endocrine disruptors, how it is going to affect the earthworm or larvae or fish like that. They'll ask. So next question with respect to ECVM 3R principle, as I told, no, alternative in vitro testing. They'll ask EC3 value calculation. They have asked. In next slide, I'll give you the example. You go through skin sensitization testis. So how this is going to affect these endocrine disruptors on the male reproductive system or female reproductive system, sperm motility, sperm, what is it, that morphology. So related to those questions, they'll ask. One question with respect to tetrahydrocannabinol and diltiazem, like synergistic additive like that, no? So this they have asked, and because of this, some metabolite is formed. Desmethyl diltiazem it is formed. So molecular weight correction factor, how we are going to do for the risk assessment? Something concept is related to this question they've asked. Then they asked one question with respect to drug study, PK effect, and co-administration there. So as I told you, it is very difficult to remember what were the exact frame of the questions. I can't remember. It is very challenging for the student to come out and remember after writing that exam for 4 hours, exact questions, the options to remember. And those questions you will not get outside also. So like that. So pause the video, go through this EC3 value, which is a measurement factor for determining this skin sensitization, concentration of the test chemical that produces 3% in ear swelling or lymph nodes. Okay. Regarding this skin sensitization also, that essay I have given in my channel, I'll give the link in the description. Pause the video, see the EC3 value, how the test chemical is exposed to the mouse ear, how actins will bind to the skin proteins, activation of the dendritic cells, lymph nodes cell proliferation, measuring the lymph node cell proliferation, calculating the stimulation index. So SI related EC3 formula they will be asking. In the options, these options they will give you, or they will give you these values, or they will ask you to give this, whether it is moderate sensitizer or extreme sensitizer like that, they will be asking. Okay, those who are working on the skin sensitization as a study director, no, they will definitely know. Other for other toxicology, it is very challenging. Stop, pause the video. Also read about blood-brain barrier. Any questions with respect to some examples and with respect to barriers, also they might ask in the exam. Okay. So as I told you, tetrahydrocannabinol as well as diltiazem, like this CYP mediated, they have asked. So you have to remember the CYP mediated enzymes will be there, no? So when it is there, no, C3A4 mediated metabolism interaction, altered diltiazem clearance, diltiazem exposure increases, it will lead to cardiovascular effects. Something related to this question they have asked in the exam. Okay, very difficult to remember everything in the after coming out of the exam. Okay, so this is the overview how the distribution of marks is there across and around 10 pages. Digitally they have given the syllabus. Most of the syllabus will be overlapping. Here in the footnote also they clearly said, see, task and they should be aware that overlap exists among all domains. They are very clearly said in the brochure also. So hope from this video you will be able to understand the few examination areas where they have asked the questions in the year 2023. So those people who are appearing for the upcoming exam in the year 2026 October. So you can go through these concepts. You can take these points and read in detail from the textbook, okay, or from the guidelines. Thank you so much. Be curious. All the best for your exam.